Buenas tardes, good afternoon. I apologize for the delay. I have some technical difficulties, but we're here right now. And Jorge is joining at this moment. So welcome to our fifth webinar of NLAAD 2022. Today is dedicated to the advances that science have done on the area of HIV vaccine. My name is Luis Alberto Mares. I am the Director of Community Mobilization Programs at the Latino Commission on AIDS in New York City. And today I will be your host and moderator for this webinar. NLAAD 2022 represents the version number 20 of NLAAD. So we have evolved and changed with the years from a poster campaign that initially just asked the population to get tested and know their status to what we now have, which is the different options to treat and prevent HIV. We have included prevention techniques like PrEP, and specifically this year, we're talking about injectable forms of PrEP and treatment as prevention, G equals G as well. And we also have included this year in previous webinars information about new treatments via injection. So we wanted to include this year again, like we did last year, information about HIV vaccine. We know that vaccines are in investigation and we soon will have more information about this. But today we will have HIV vaccine research updates with Jorge Benitez from the Columbia University of Research Department. Jorge is right now logging in. As I mentioned, we have some technical difficulties, but we solved them together and he's joining in a minute. This webinar is being recorded and we will post this on social media. And please, we will have a presentation by Jorge, and at the end, there's already time for ask your questions. So I suggest you and ask you to please write your questions in the question and answer box. Are you logging in, Jorge? Okay. A little bit about Jorge. Jorge Benitez is of Ecuadorian and Puerto Rican descent, who grew up in East New York, Brooklyn. He has invested 20 years of his life to research and potentially highly effective prevention of HIV. 16 of his work has been dedicated to efforts to speed the development of HIV vaccine. Key populations with whom Jorge has worked among include, but not limited to, HIV negative and Latinx LGBTQIA+ people, cisgender people, and drug users in efforts to decrease transmission within these particular populations. Jorge has also participated in NLAAD in previous years, organizing testing events, and I found that in my records, Jorge. And he is very excited to share about cutting-edge HIV vaccine research with you all today. No, he's logging in right now. So, okay. If you were present last year, we had a presentation of HIV vaccines with Jensen last year. And unfortunately, when it's presented by a laboratory, pharmaceutical laboratory, we cannot record or share the slides. And that's why there is no records of that webinar. But what I remember is that what was presented is that there have been several attempts to have HIV vaccines since a long time ago, but it's been a difficult process, and many studies and research did not succeed. So Jorge is going to tell us today about where are we in terms of HIV vaccine. I think we all want a vaccine to be out of the market as it happened with COVID last year, and we were able to stop the COVID spread. So we will be very happy to have a vaccine of HIV and be able to stop this epidemic faster than we're doing it. But we're also in the process of ending this epidemic. It's been a plan that has been published first in New York State and then in many other states and the end in the entire country to end this epidemic by 2030 with the tools that we have now. And that's what NLAAD has been promoting from the very beginning because we know that people need to be tested because 15% of the people living with HIV do not know that they have HIV, and those people who don't know unknowingly can spread the infection. But we have tools now like units, for example. So when we have these people identified, they should start treatment as soon as possible so they can reach viral suppression immediately, as faster than in the past happened. So people who reach viral load lower than 200 sustainable are not capable of transmitting the virus to anybody else. And this proved scientifically. So from the 80s, but we have no cure, treatment, or anything, to what we have now, it's been a lot of advance in these 40 years of epidemic. One more thing that we would like to have in our arsenal of tools to end the epidemic would be a vaccine. That would be great. So as I mentioned, Jorge will present on that today. Yes, Jorge. I saw that you are logging as a participant, not as a panelist. That's quite probably. You cannot. I asked you in the chat to unmute yourself and. Okay. I send you a promotion as a panelist. Okay, perfect. You are now.
Perfect.
Yeah. All right, we are here. Thank you. I'm apologize for the inconvenience of difficulties, but we are here now.
No worries. And if Luis, are you able to share my slides?
I looked for them. I didn't receive them. Check if this. I didn't get them. You can send it to me, and I'll do that.
Yeah, I did this morning. You should have it at 11:52. And make sure I have your correct email.
It's Elmares? Elmares?
Yeah, Elmares at latinoage.org.
Yep, I don't have it.
Huh. Okay, how about I'll just do it from my end. Can I share screen?
Yes.
Okay. Oh, here you go.
All right.
I'm gonna have to do this with notes.
Yeah.
I can't do that.
Uh-huh.
I'm gonna have to go back because I'm gonna need notes. Okay. Hello, everyone, for joining the call today, and happy National Latino AIDS Awareness Day, which is actually tomorrow.
Tomorrow.
Yeah.
No, we have the office. Happy NLAAD.
And thank you all for joining this call. As Luis mentioned a little bit earlier, what I'll be doing today is sharing. Obviously, there's different ways that we can help prevent HIV and even treat the infections. And with the National Latino AIDS Awareness Day, we're focusing on how we can do this among our Latino community. And today, I will be sharing about work that we're doing to help prevent HIV by developing HIV vaccines. We've had many successes with vaccines in the past. Very recently with the COVID pandemic, we've had many successes in being able to control infections and through the use of vaccines that were disseminated in the past couple of years. And we hope, and we've been doing the same research in efforts to prevent HIV as well. Actually, all of the research that was done with the COVID vaccine effort was built on the backs of the work that we had been doing with HIV vaccine research. Many of the vaccine research sites, of which there are over 40 sites on about 5 different continents, have been converted to COVID vaccine clinical site research sites. But both viruses are very different from each other. And so we've had specific challenges with finding or developing a vaccine to prevent HIV. So at Columbia Research Unit, Columbia Research Unit is a research group that's funded mostly by the NIH. We are part of a couple of research networks, especially clinical trials research networks. The unit was first designed and developed or established over 20 years ago, and the main focus of our work was initially to treat HIV infections, and then a few years after the focus of HIV treatment, then we began testing HIV vaccines. And we've been doing other research as well, such as I mentioned, the COVID vaccine. And we are also now conducting a study to treat MPV, also known as monkeypox. And our research unit is made up of community engagement staff, so people that educate and conduct recruitment for studies that we conduct. We have research assistants that conduct some of the clinical procedures in our clinic. We also have clinicians that are made up of nurse practitioners, doctors, and physician's assistants. The lady on your bottom right is our principal investigator, Dr. Magda Sobieszczyk, she's the chief of infectious diseases at the New York Presbyterian Hospital in Washington Heights, and also our principal investigator. And then on your upper right hand, you have 2 coworkers that are doing makeup before New York City Pride. So part of our motto is if you do makeup together, you can help prevent HIV together. So as far as the work that we're doing, we still there is still an issue with new infections. There's still about 37,000 people that get HIV each year here in the United States, and out of those people that get HIV annually, about 27% of those that get HIV annually come from Latinx populations. So that's about 27% of the people that get HIV. While we as Latinos and Latinx individuals only make up about 16% of the U.S. population. So there are, as far as percentages, there are more Latinx that are getting HIV as compared to other races and ethnicities in the country. And what could an HIV vaccine do? So basically, vaccines can do a number of things, and you've heard about this recently too in the last couple of years as it related to COVID vaccines. So one of the things that we know is that a vaccine can actually prevent infection, right? There are many people that have gotten the COVID vaccines, for instance, and did not get COVID as the result of getting the vaccines, and probably likely also being careful about how to prevent an infection by washing hands, keeping hands away from the mouth, and wearing masks. But we also know that even if you got a COVID infection, a COVID vaccine, that there was still the possibility that you can get COVID, and this is something that happened over and over again as well in the country. However, one of the benefits of the vaccine has also been that it delays the disease. So even if people got vaccinated for COVID and got COVID, they were likely not to have to go to the hospital, and because their disease did not progress to something that was too much to handle. Other benefits of a vaccine could be that it reduces community transmission. Well, speaking about COVID again, or using that same comparison, there was one point where here in the city, we were keeping an eye on, and also different parts of the country, but we were keeping an eye on how many people got the COVID vaccines. There was a number that we were shooting for as a goal, so that we could reach what we call viral immunity. I mean, community immunity, and that number was about 70%. So researchers and scientists felt that if about 70% of the people receive COVID vaccines, then it would also help protect people that did not get the vaccines. So how do vaccines work? Essentially, I was hoping to have somebody answer this, but essentially what vaccines do, the body recognizes the proteins and the genes that are in a vaccine. Then once the vaccine or the person is inoculated or the person received the vaccine, then the immune system sounds an alarm because it identifies these new antigens in our body, and it wants to know more about them and wants to get rid of it. And so once that alarm is sounded, we have parts of our immune system that go into action, and basically there are fighter cells and proteins that go into action to eliminate or neutralize the virus. That is the actual goal, to kill or to control it. There are a number of strategies that scientists have used over the decades in being able to develop vaccines. So like, for example, common vaccines such as the chickenpox or flu use a whole or weakened form of the virus. There's other vaccines such as the Hep B vaccines that only use safe pieces of the virus. However, in the HIV vaccine studies that we conduct, we don't use any whole or weakened form of virus. We just feel that would be too risky to use it as part of a vaccine strategy. And we also, from collecting feedback from community, the community would probably not want to get a vaccine that uses a whole or weakened form of the virus. So we develop vaccines and test vaccines where there is no real HIV in the actual in the vaccines that we're testing. So there's no way possible that that someone could be infected by HIV from a vaccine that we're testing. I'm just gonna, but okay, so here we have a slide that illustrates how memory works. So if I were to have, if I were to show just these pieces of the image, I think almost everyone that's in the webinar right now would be able to recognize that these are all parts of a car. However, if we were to put these parts on a table or try to put them together, you would not be able to drive away with the car, because there just aren't enough pieces or enough functional pieces to make this car drivable. And that's sort of the same strategy that we use when we develop and as we test vaccines. We are using only pieces of the RNA or the DNA, but in this case, it's the RNA of HIV because it is an RNA virus. We use very much like in COVID, which had a spike protein. There are similar proteins on HIV as well. And we use some pieces of that as well. But we know that there aren't enough of these copies of pieces of HIV to make a functional virus. And we, just like your memory connects these pieces of the car to, these pieces to a car, we hope that the immune system will recognize these pieces as pieces that belong to the HIV virus. So, when scientists put together a vaccine, and they have an idea for a vaccine, is not like those vaccines just begin to get tested among people right away. They go through a very stringent research process that includes testing the idea in laboratories, and then the same vaccines that we have hope that can benefit people are tested among animals. And then we look for, in the animal model, we look for symptoms, we look for any what we call adverse effects, meaning symptoms that are bad. And before a vaccine can move to be tested among people, the data and the research has to show that these vaccines are safe, that the animal model can tolerate the vaccines itself as well. And then we also begin to see in the blood work that we draw from the animal model that these vaccines are also immunogenic, meaning that the vaccines are helping to induce an antibody response. But when the vaccines get tested in people, we generally test the vaccines in 3 main phases of research: phase 1, 2, and 3. Generally, in the phase 1 and 2 type of research, these are studies that include very small number of people. So in a couple of slides, I'm going to introduce a study that we're conducting right now, which is in the phase 1 of research, and which includes 108 people. In a phase 2 study is conducted once we see that the vaccine in the phase 1 is safe, people can tolerate them, and we begin to see in the blood draws that the vaccines are helping to induce antibodies, then it will move into a phase 2 study. But there are times that we and scientists are so convinced that the vaccine is safe and tolerable in the phase 1 level of research that it jumps into a phase 2B study. And in this type of a study, we enroll people, we enroll 7,000 people into this type of study. And we also enroll in the phase 1 and phase 2, we enroll people that are at low risk of getting HIV. But in a phase 2 and a phase 3 study, we begin to see if the vaccines work by enrolling people that are already naturally are showing some behavior that may potentially put them at risk of getting HIV. And that would include having multiple partners and wearing condoms and with very low frequency. And these are folks that we would want in these types of studies. So as I mentioned before, these study vaccines are experimental, and they definitely cannot be found at the community level. Like they cannot be found in drugstores, pharmacies. They cannot be found at your doctor either. As I mentioned before, that the study vaccines definitely do not cause HIV. However, there is a chance that that the vaccines that we're testing may decrease, it may increase, or it may not even change your chances of getting infected with HIV. I've been doing this work for about 16 years, and somewhere like around 2009, we had a small phase 1 study. Actually, we had a couple of studies that were using a component that's called an adenovirus. It's essentially a cold virus, and we were inserting the copies of genes and proteins into that virus, into the cold virus, and then we would inject the participants with that cold virus. The strategy or the hope was that the immune system is going to respond to the cold virus. It wants to eliminate it, and then if once the immune system goes to fight off that cold, then the cold virus will present the copies of proteins and genes of HIV. But it was found that if there was a combination of things happening, such as previously having been exposed to that cold virus, and for men, if men were uncircumcised, and if there was a combination of these two characteristics, those characteristics were putting some people at high risk of getting HIV as a result of getting the vaccine. Since then, we have stopped using that adenovirus as part of the vaccine strategy, and that is something that we definitely learn not to do as we move forward. So I want to dissect one of the studies, as I mentioned a little earlier. The study is called 302. 302 is in a phase 1 level of research. We just recently fully enrolled the 108 participants into the study just a couple of months ago. But one of the neat things about this particular study is that it uses the mRNA vaccine strategy that was also successful with the Mrna vaccine to fight COVID. And we are, even though we have COVID vaccines that are already approved that are using the mRNA strategy, we need to begin sort of from the beginning with the HIV vaccines by collecting data just to make sure that the HIV vaccine using the mRNA strategy is safe and can be well tolerated. So it's right now we're in the taking baby steps. The study is about a 12-month study. So in next summer in 2023, everyone in the study would have completed their participation, and then we begin analyzing the outcomes of the vaccine. And some of the research questions that we generally ask in all phases of research are the study vaccines safe to give to people? Are people able to take the study vaccines without becoming too uncomfortable? And if the vaccines are immunogenic? And then but in the phase 3 and the phase 2B study and the phase 3, we also ask the question, is the vaccine effective at preventing HIV? So just some honing in on this particular study, the 302 study, there are actually 3 different vaccines that are being tested within this protocol. Each vaccine is slightly different, but they all use the mRNA strategy. And the mRNA strategy this is sort of like the cycle. So first, the mRNA that has the instructions for making pieces copied from HIV enters a human cell, and then the mRNA in the cell delivers the message to make pieces copied from HIV, right? So, and then the cell makes the pieces and displays them on the surface, as you see here. And the immune system sees that piece and recognizes them as foreign and creates an immune response. It sounds simple, but it's there's a lot of chemistry or organic chemistry that goes on within those cells. And want to share that. So just like many of you have experienced symptoms when you've gotten your COVID vaccines, people may experience those same symptoms if one were to participate in a vaccine study. So I feel like all of us know of, if not ourselves, or know someone that experienced one or more of these side effects when receiving the COVID vaccine. And this is again true for the HIV vaccines. One of the reasons why why people experience some of these symptoms is that the vaccines are actually integrating themselves with the immune system, and as the immune system responds to the vaccines, some people, if it's a greater response, you may feel more of these vaccines than someone whose immune system doesn't respond as well. And again, so these are the same risks for the study vaccines, but just to reiterate that we don't know all of the risks yet for the vaccines that we're testing within 302. This is something that we'll find out within the study. And then the 302 study is divided into a few groups. But this is a chart that actually shares the injection groups. So in this study, there will be, if someone is in groups 1 through 6, they'll receive 3 injections of the vaccines. There are going to be some people that will receive a lower dose of each of the vaccines that I mentioned before. And then there are going to be some people that will receive a higher dose of the same vaccines that I mentioned a little earlier. This is also important to try to measure because we want to also find out how much vaccine we actually need for to effectively prevent HIV. And we also recently experienced the importance of having differences between higher dose and a lower dose of the vaccine with the monkeypox vaccines. So there was at one point when we had a stock of monkeypox vaccines, each vial that we had for monkeypox was for one person. However, during the monkeypox outbreak, it was found that researchers found that if we use a fifth, like 20% of the vial, and instead of injecting it in the arm, we would inject it subcutaneously. We found that it could be just as effective at preventing monkeypox as using a whole vial. So that was able to stretch the amount of vaccine that we had to serve more people than we would have if we only used the higher dose of the vaccine. And in 302, participants will attend about 11 visits over 12 months. Again, 3 of these visits, folks will get injection of the vaccine, right? So this would be at the very first study visit. The study begins with the first injection, and this one would be 2 months later, and then 6 months after the first injection. But in between study visits, there will be some follow-up visits where we monitor health. We assess how well the vaccine is doing, and we also check in on the participant just to check in on comfort level, making sure that everything is okay with the participant as best as possible. And one of the things about the research as well is that sometimes we have to randomly assign participants into different groups that are in the study. And we do this sort of like flipping a coin, but something that's very important is that the participant never gets to choose which group that they're in, and nor does the clinical team or the study team. So this is all done randomly, usually through a computer. Now the study also has something that's built in. That's called, we have what are called study pauses. So essentially, when I mentioned these groups over here, so the study is designed so that whenever there are 4 people in each of these groups that are enrolled into each of these groups, after the 4th person is enrolled, the group will go into a pause. It'll go into a safety pause in which there's something that we call a safety data monitoring board. This is a group of people that oversee the research to to make sure that the research is being done ethically and in a safe manner. And so, yeah, the study has a pause, and then the folks from the safety data monitoring board, they assess to make sure that the vaccines are safe to continue, and if they are, then the study, the pause is lifted, and then we can continue enrolling into the study. However, if they see that there is a pattern of people potentially not being safe while in a study, then the safety data monitoring board can make a decision to stop the study from continuing. And something that's important about the safety data monitoring board is that it's an independent body. They do not work with Columbia. They do not work for the NIH. They are scientists and researchers that are focused on that role. So preventive vaccines, again, cannot cause HIV infection. There is an informed consent process that explains the study in detail. We have consent forms that are about 20 pages long that essentially explain the study as best as possible. And to date, there have been over 30,000 people that have participated in HIV vaccine trials and have had very low, very few side effects. To be able to join a study, we will find out if you're eligible. And to find out if you're eligible, one of the key things that we do is we ask people to complete a physical exam with us, where we check in on kidney health, liver health, hepatitis B, hepatitis C, and we also conduct STI screening. One of the things that can happen, just like the COVID vaccines, if we were to go to a lab, we can actually request labs that can help identify if we have had COVID vaccines, and this is the same is true for the HIV vaccines that we're testing. Now, if folks in studies were to receive an HIV vaccine that we're testing, there's the likelihood that the person will experience something that we call VISP. VISP stands for vaccine-induced seropositivity, and that means that that an HIV test result can come back antibody positive if someone were to test for HIV after they've received an HIV vaccine. In the laboratories that we work with, when participants in studies get tested for HIV, what the laboratory does is they test the blood work to find if there are antibodies. If there are antibodies, then they take that same blood work and look for virus. What the participant gets back as a test result is whether it would be one of two things. It would be HIV uninfected or HIV infected, but it's based on those two types of tests. And to help facilitate the fact that some people have this have VISP, we provide lifelong testing assistance for people that have VISP. So one of the key things, like for instance, is if you are in a vaccine study, when we test your blood work, it comes back antibody positive. You would be eligible to get to be part of an HIV testing services that are part of the HIV vaccine trials network for however long you may need it. It could be for whenever the antibodies wear off, or if they don't wear off, you can continue to get tested with us. So getting tested with us can be in a number of manners. One, you can go to the clinical research site where you had tested before or where you participated. if you moved away, you can transfer or get tested at one of the clinical research sites that are in the country. Or if you happen to be someplace where that you can't access a research clinic, what we would do is at the end of the study, we will give you information so that you can keep in touch with our clinic, and we will mail you an HIV testing kit to your home. And then all you would have to do is essentially go to a Quest Diagnostics, which can be found in almost any city in the country, and also other parts of the world, so that you can get tested for HIV. Study participants will also get reimbursed for joining studies. We compensate for your time, your travel, the inconvenience for being in the study, and generally for visits. Going back to this slide, for visits that require an injection, folks will get $100. But if the visit is a follow-up visit where you would essentially just have blood drawn, you will be compensated $50 or $75, depending on how many tubes are needed for the visit. It's generally about up to 6 or 7 tubes. It's $50, and anything beyond that is $75. So I wanted to talk. I usually do this presentation with my coworker, Jorge Soler, who works at Project Achieve. And Project Achieve is also a partner of Columbia Research Unit. We're basically joined at the hip conducting research. And so these are some slides that Jorge have put together for us. So I want to share about who participates in HIV vaccine studies generally. So the HVTN conducted an analysis of people that we engaged into studies between 2002 and 2006 that included 43 studies at 18 sites in 14 different cities. Nationally, Latinx made up about 8% of study participants, while Latinx make up among 20 or 19% of the US population. It's actually a little bit more now. Our goal is to match the percentages of the people enrolled in HIV vaccine studies to the percentages of the US population. So for us, that means that would be a proper representation. There was also an article that was published by my coworkers and me in 2015 that highlighted the representation of Latinx and Blacks into HIV vaccine research. And we basically looked at screening and enrollment data from 2009 to 2012. So when it comes to engaging people into HIV vaccine studies, we essentially have to engage many people. So we have to engage 6,777 people just to get 96 people to enroll into one of the studies. And this was, again, from that time, from 2009 to 2012. That translates to about 63 people that we have to engage, or 63 people that are interested, meaning that they provided their contact information because they want to know more or potentially want to participate, for each person that winds up enrolling. And if you see here, the slide on the right essentially illustrates, if you see that the zone is smaller and as it gets bigger, that means that the percentage of this group is increasing from the time that we recruited them to the time that we enroll them. So in other words, of focusing on here, Latinx have made up about almost 36% of the people that we engaged and that provided contact information. But by the time that they get to the enrollment, Latinx only have made up about almost 18% of the enrollee population. So from contact to enrollment, the participation of Latinx has dwindled by about 20%. And this is a slide that also shows that we engage, if you see these numbers on the top, we engage more Latinos into HIV vaccine studies as compared to whites and blacks. But Latinx happen to make up the lower percentage of people that actually enroll into studies. And these are some reasons why some people may actually drop off out of studies. So there's some things that had a higher drop-off rate than other racial and ethnic groups. So things of having the adenovirus that I mentioned before. This was back in this time during the 2009, 2012, we were still testing adenoviruses, but we were just at the tail end of not testing them any longer. There were some studies that also required male participants or people that were assigned male at birth, so there were higher circumcision rates among Latinx men as they were to the other races and ethnicities. So fewer Latinx could have participated because of those, I guess, those conditions. And also, just diving in a little bit more on why people do not participate. So some barriers to participation have historically been HIV-related stigmas and fears. So many people decide not to join studies because of the stigma being connected to an HIV vaccine clinic and the stigmas of HIV under those circumstances. Some items that actually have facilitated participation of some individuals were the patient felt that the vaccine had some sort of efficacy or were hoping for some level of efficacy. People had strong beliefs about clinical trial protocols as well, which is like the highest reasons. But hope is in the air. There are 2 studies that have given us hope in the past 10 years. So in 2012, we received results for a study that was conducted in Thailand that involved over 16,000 participants. And it was found that particular vaccine had 31% efficacy. The study actually needed, or the vaccine needed, at least 50% efficacy for it to be licensed. So it fell just short of being able to be licensed. However, that strategy of that vaccine has been tweaked, and it is now being tested in different continents, including Africa, especially in sub-Saharan Africa, the United States, South America, and Switzerland. And then there was also another study where we were actually skipping the step of a vaccine. So instead of developing a vaccine so that we introduce it to your body and then your immune system responds to the vaccine by by developing antibodies, we also conducted a study where we were testing broadly neutralizing antibodies that were engineered in labs. And we were infusing these antibodies directly into study participants. And in this study, which was called the AMP study, which is AMP stands for antibody-mediated prevention, there were over 4,000 people that joined, and that study showed a 30% overall efficacy as well. But there were, so this strategy was hoping to be able to prevent infection from different types of, or different strains of HIV. And these antibodies did have higher levels of efficacy on some specific strains of HIV. Okay, so please follow us if you want to know more about Columbia Research Unit and the research that we're conducting. You can follow us at any of these handles, Facebook, Instagram, Twitter. Want to give thanks to the people that contributed to these slides. More importantly, I want to thank all of the participants that have helped with the results that I was able to share with you today. There have been over 20,000 people just in the last 2 studies that I mentioned. And I also want to thank my coworker Jorge for letting me borrow his slides. And thank you to you all for hanging in there with us with our technical difficulties.
Thank you very much, Jorge. We're going to proceed with the questions. There's 3 questions, but it was mentioned that you worked in this field for 20 years and you were with vaccines for 16 years, I think. And how many trials, studies have you been part of, and how or do you know about since you started working on this? It's many, right?
Yeah, I would say overall within our research network, we have tested over 70 different vaccines. Most of the vaccines that we tested were in the phase 1 or maybe the phase 2 and never got much further than those 2 phases of research. But in the phase 3 level of research, or 2B, where we were finding out the efficacy of the vaccines, we've tested about 8 globally thus far.
Like 2 years ago, we were contacted by, I can't remember exactly who, probably a Fresh Hatch. There was a study that was being done in different countries called Mosaic.
Yes. Yes.
Do you know what happened with that? Because it was going to be released some information and then nothing happened.
Yeah, great question. So the study that I mentioned a little earlier, which is the RV144 study that was done in Thailand, that was a study that had the 31% efficacy. And that particular vaccine was changed a little bit, tweaked a little bit. One, so that we can test it in South Africa and in the North and South America under the name Mosaico. And the Mosaico study was fully enrolled sometime in the summer of last year, and we expect for the study to be completely finished by the summer of 2023, and that's when we'll begin. Unless between now and then there's some data that is very strong and leading, to say that the vaccine is working or the vaccine is not working, we'll start to get results sometime after 2023.
And these studies are part of global.
Yes. So there was an arm that was conducted in over 30 sites in sub-Saharan Africa. It was done in North America. There were about 22 sites here, down in Peru, Argentina, Brazil, and in Switzerland. I may have missed one country or 2.
Yeah, I remember you mentioned because it was Mexico and Peru. I remember that. Mm-hmm. Is there a way that the people who start these studies or these projects decide where to go? Why Mexico? Why Peru? Why Sub-Saharan Africa and not Europe, for example, or China or other countries? Is there a factor?
Yeah, so we're following where the epidemic is impacting communities the most. Sub-Saharan Africa is the part of the world where HIV is impacting people the most. And then there are countries like Peru, where things are happening in much the same way. Like, for instance, in Peru, the government, I don't know about right now, but while we were conducting the study, the government was not ready to disseminate PrEP. and the country did not have an overall access to PrEP. So if you don't have access to PrEP, then your risk of getting HIV is probably a little higher, right? So we were working in communities that were most affected and at risk of getting HIV.
Interesting. We are already in this epidemic for 40 years in the United States, probably longer in the world. Why do you think it's so difficult that we in these 40 plus years haven't found any vaccine yet? When for COVID was, I mean, we know that we use the technology that has been developed for HIV vaccine into the COVID vaccine and that worked. But why we haven't been able to get HIV vaccine yet for so long? Hello? You're frozen. Oh, he will join again probably. Let me see here. I wish I could answer your questions. The question of Francisco Diaz says, is it true that people who have participated in prior HIV vaccine trials cannot participate in future trials? I think the answer for that question was given through the presentation with the brief explanation, right? That people who participate in trials, they develop some sort of reaction that is going to look like it's positive. So therefore, we'll not be able to participate in a different trial. About the funding, probably we'll have to wait for Jorge to rejoin. Also with exclusion criteria, I don't remember exactly, but after you write this question, I think Yuri, Jorge explained the factors or the details of who were elected to participate. And he was about to answer the question that Yuri asked also, what is the challenge to move trials along, participation or efficacy? The combination of both or more factors. Let's wait a little bit to see if Jorge join us again and is able to answer the questions. One thing that I was going to ask Jorge is why is it important that we enroll Latinx members in these studies? As you know, the Latinx Commission is also run the serious pandemic, which is done in combination with the COVID-19 Prevention Network. And we started that series because in 2020, when they started to make the studies on HIV, COVID-19 vaccines, the rate of participation by Latinos was very low. And in order for the studies to be able to produce a result that we can say 100% sure that it's applicable to every community or every part of society, every part of society and every community needs to be represented in those groups of studies as well. That's why it is very important that our community, Latinx, be part of the studies. And we were trying to do that and promote that back in 2020 for COVID-19. And I think that's what Jorge also presenting for HIV. Well, I will transfer your questions to him because I don't think he got frozen probably, so I have technical difficulties again. So thank you very much for all of you for your participation, and we will be back with our last webinar on the 20th in a week about you equals you, and why it's important that we also disseminate information in the communities because we are all in this job in this task of ending the epidemic. And all these tools that are being presented in these webinars are very important for us and for everybody to end this in this country. Oh, well, thank you very much again for your presence here today. Apologize for the difficulty at the beginning and at the end of the webinar. And hopefully this won't happen again. And see you next time. Bye-bye.